Okay, everyone. Thank you so much for coming. I'm really excited to have the opportunity to introduce Sara Walker to you today. She is an astrobiologist and a theoretical physicist. Currently, she is an associate professor at CC at ASU, the School of Earth and Space Exploration. She's also the deputy, this list is going to be long, so strap in, guys. She's the deputy director of the Beyond Center of Fundamental Concepts in Science. She is the associate director of the ASU Santa Fe Institute Center for Biosocial Complex Systems. She's also the co-founder of SegaNet.org and on the board of directors for Blue Marble Space. I could keep going, but I'll stop there so that we can actually listen to her talk. Sara did her PhD at Dartmouth in physics and astronomy in 2010 and a bachelor's in physics at Florida Tech. She likes to think about questions in the origin of life, thinking about information in living systems, planetary networks, and identifying life on other worlds. So today we're going to hear a lot about her work, specifically in planetary systems and the laws of life. So please join me in welcoming our speaker.
Thanks. Great. Can everyone hear me okay? Excellent. So I'm really excited to be here today. It's been wonderful interacting with people this morning. I'm excited about more conversations this afternoon. What I want to talk about today is some work that's been being developed in my group over the last few years, which I'll get into in a few minutes. But I always like to give a little bit of background about my perspective and philosophy on the way I do the science that I do, because I take a bit of a different perspective than most people in my field. So I am an origin of life scientist, but I was trained in theoretical physics, and so I'm very interested in deep principles and whether there are actually laws of life, which is in part inspiring my title. And in that sense, whether there are actually universal principles that govern living systems, not just on Earth, but anywhere in the universe. And so having been trained in cosmology, I was very familiar with these kind of diagrams, which probably most of you are. But as an astrobiologist, I'm actually really interested in a missing fraction of the universe that we don't understand yet, which is how much of the matter that we actually have actually transforms into living systems. And so this is sort of like the framing of the way I think about the problem. There's a long tradition, actually, of physicists that have been really interested in thinking deeply about what is life. And Erwin Schrödinger is probably the most famous, so a lot of people will cite him, and in particular, his book, What is Life?, which inspired a lot of people to think about life from a more fundamental perspective, including Watson and Crick, to look for the structure of DNA, which Schrödinger actually predicted. But the thing that I think is really interesting is just the way he asked the question. So he was interested in whether the events that can happen within a cell could actually be explained by physics and chemistry. Right, so we have pretty good theories in physics and chemistry that explain the natural world, but we haven't actually come up with an explanatory framework for life. And so what Erwin Schrödinger did in this book was actually go through a logical set of reasoning about what the properties and rules of life might be based on the constraints of what we know of physics, which is one of the reasons he could predict DNA had to be an aperiodic crystal, because it needed to actually conform to what we knew about crystalline structures, but it actually had to have a high algorithmic information content. So he basically did this kind of logical deduction. But what he ended up coming up with at the end of the book, which I think is actually probably the most insightful thing he said in the entire book, was that living matter, while not eluding the laws of physics as established up to date, is likely to involve other laws of physics. So he couldn't explain life in terms of like entropic principles or any of the kind of things that he was actually really thinking about coming from the perspective of a physicist. And he thought that probably some new principles are going to be necessary to explain life. And so I actually take this as a point of inspiration. I do think that we have missing principles that will eventually explain life, but we really don't have any idea what they are yet. And so this motivates most of my work. I'm interested in what those fundamental principles are. And if I think about where we've gotten sort of in physics in general, we've got like sort of this series of unifications that have happened and gotten us to an ever deeper description of reality. So we have electricity and magnetism were unified in the 1800s, and then we got electroweak force in the mid-1900s. But I think that this quote from David Krakauer, who's president of the Santa Fe Institute, is actually really telling about where these theories sit in our understanding of the world. That this goal of a theory of everything that physicists have is a theory of everything except the things that theorize. Right? And so part of the motivation for that is actually that we have this whole world of the complex and all the kind of systems that we interact with, technological systems, living systems, that are completely missing from this picture. And so when we're talking about fundamental descriptions of reality, we don't include ourselves in those at all. And so this has actually been a deeply problematic thing for theoretical physics for a really long time, that we don't really understand observers or what we are as observers. And John Wheeler, who's a really famous theoretical physicist, had this great dictum, it from bit, where he talks about all things physical should be information theoretic in origin, and this is a participatory universe. But basically this idea that maybe there's something about information that's missing in our descriptions of reality, and maybe information actually is something that should be fundamental. And so this guy gets into thinking about some really deep things about the classical and quantum world and different descriptions of things. But what does that have to do with life? And how do we actually go from thinking about those kind of problems to thinking about life and the physics that will describe life and understand what that kind of physics is going to look like? And so one of the questions you might ask is, what is information, and what is information doing when we're talking about information, and in particular, information and its role in biological systems like us, right? So Wheeler was talking about scientists and scientists using information to describe the world, or information being fundamental to the world. But one of the ways that I like to think about information, because a lot of people think about it really different perspectives, is just to think about what biological systems do with information. And my favorite example of this is actually to think about the Earth and its anti-accretion that we're undergoing right now. So a lot of us are probably familiar with the idea of planets accreting mass, right, in solar system formation and things. We actually have planetary formation models via accretion. David Grinspoon has this nice analogy where he says that our planet is the only one that's anti-accreting. Basically, we're launching little metal boxes into space. Why can our planet do that? Well, our planet can uniquely do that because we have a technological civilization, us, on the surface of the planet. That technological civilization has a certain kind of information. Knowledge of the laws of gravitation, right? And knowledge of engineering principles. And we use that as a way of mediating certain transformations, this anti-accretion process. And so one of the arguments I like to make about information is required for these kind of physical processes that wouldn't happen without information, which is kind of tautological. But basically, you wouldn't end up getting a planet anti-accreting unless there was some kind of biological, technological civilization that actually had information about that kind of physical process. And most of what biology is doing is accruing that kind of information to mediate transformations that wouldn't be happening in the absence of actually having that kind of knowledge. Another example I like to give is to think about technology and its place in sort of the periodic table of the elements. So you'll often see the periodic table with all the different kinds of processes that happen in the universe as far as the physics and what generates those elements. But once you get into the highest mass elements, you'll notice that they're only produced by technology with any abundance, right? So it actually seems to also be a case where some kind of knowledge, some very deep information about the way the world works, about nuclear physics, is actually necessary for some of these elements to exist. And so, so this seems to suggest, at least to me, that there's some really interesting physics that biology does, in the sense that biology allows certain processes, via its use of information, to occur that wouldn't happen otherwise. And this is actually nicely captured by David Deutsch, who's a physicist at University of Oxford, where he talks about the fact that base metals can be transmuted into gold by stars, and by intelligent beings who understand the processes that power stars, and by nothing else in the universe. And so when I talk about laws of life, or this idea that there's missing physics that's necessary to describe life, what I really mean is that there's some basic physics, like the fact that we know metals can be transmitted into other metals. And then there's a new kind of physics that we need to understand, where there are actually some physical systems, some parts of matter, material reality, that understand those processes and can actually control those transformations. That's us. All right, I want to understand that physics. What is that physics? So I'm going to get into things that are a little less deeply philosophical and a little bit more concrete now. But the main reason for this kind of philosophical introduction is that I think when people are thinking about defining life or understanding life, they're not thinking necessarily about how conceptually deep that question actually can be. I think we have a lot of sort of phenomenological descriptions of life, but they're not really at the level of having quantifiable theory. And so one analogy I like to make in terms of how I think about this is, like, if we think about gravitation, we're all pretty familiar with the fact that we're stuck, to the Earth, and if I jump up, I fall back down, right? But it took some really deep insights from Einstein and others about the nature of the curvature of space and time to really figure out an explanatory theory for that, right? And life is so much more complex than gravity. That there really must be some kind of really interesting physics that's governing living systems. So one of the proposals that I've come up with various colleagues, in particular Paul Davies, who is my postdoc advisor, is the idea that the origin of life transition itself is actually a transition that would be best described and quantified potentially in terms of an increasing role of information in structuring matter. And so an obvious example of this is the fact that biology uses DNA and other digital polymers for mediating information. But I would say that the structure of chemistry as it occurs in biology is completely governed by information actually mediating that structure. And so one way that we talk about how you might quantify the origin of life is actually to think about it as a transition informational structure of matter, and that we might actually be able to get at some mathematical principles governing that. And so the argument I really like to make is that life is not physics as we know it, because information determines what transformation happened, not just the laws of physics alone. And I want to understand how that process emerges from random chemistry. Okay. So that ends sort of the philosophical stuff. I'm going to get into much more sort of rigorous quantitative science now. But I wanted to at least give a framing of like where my perspective comes from on things, because I do have a tendency to ask questions quite differently because of that framing than people traditionally do. I'm an astrobiologist, so I'm interested in what makes this planet alive and this planet not alive. And so just in the context of the discussion that I've sort of given so far on the role of information, one of the arguments I would make is some people might say that what's happening on the surface of this planet is just a process far from equilibrium, and we just need to understand far from equilibrium physics to understand cities, right? But if you have something like the Great Red Spot of Jupiter, that's also a far from equilibrium process, and it's a self-organized one. And so what's the difference between the Great Red Spot of Jupiter and a city? Right? I would say that the difference is that a city requires algorithms and information to construct it, and there's a lot of actual, you might call it intelligence or knowledge in a city, that's not necessary for the Great Red Spot of Jupiter. So I'm going to come back to thinking about planetary scale life and how we can detect it at the end, particularly with exoplanets. But right now what I want to do is kind of move into chemistry and think about these sort of abstract ideas, but now how do we root them in understanding what are the differences between a living chemistry and a non-living chemistry? And could we actually quantify those differences? Okay, so living systems, are they actually different within the space of possible chemical transformations? So we know some things about how we can think about system level properties. And so I know Carol Cleveland was here last week and also talking about defining life is my understanding. So I really love one of her papers, which this is actually just totally coincidental that I had this slide up and then heard she was here last week this morning. But she has this great paper on defining life where she talks about the analogy with water and the fact that you could try to define water in terms of its obvious properties, like it's a clear liquid, it's a liquid at room temperature, but you wouldn't uniquely define water. The only way we actually uniquely defined water was once we had a proper molecular theory, and we could actually identify it as H2O. So the argument that she was making in that paper with Chris Chiba was basically that we need to have theories, not definitions. And I'm very much in that camp. But the interesting thing about water is itself also an emergent phenomena. So even if we talk about water as H2O, the properties of water that we associate with water actually require thousands of water molecules. And in fact, people have tried to precisely calculate how many water molecules it requires for water to be wet and have the properties we associate with it. But nonetheless, it's actually an emergent property. Water itself is an emergent property of many interacting atoms. We might also talk about life as an emergent property, but it's an emergent property of many interacting molecules, right? And so when we're talking about what defines a living system, a living chemistry is different from a non-living chemistry, it must be something in the structure of the patterns of the interacting molecules. And so a lot of people in astrobiology are very concerned with the specific components life is made of, like amino acids or the sugars in DNA or lipids. What I'm interested in is what are the patterns in terms of the transformations in that chemistry. In part because that's a little bit more agnostic to the particular chemical details and therefore maybe more universal to life on other planets, but also because it is very consistent with this idea that life is really about controlling the transformations that happen that I was describing earlier. And so what we want to do is basically try to characterize the patterns that are in living chemistries and then try to develop some theory from that to basically explain what life is or understand what life is. So right now in my group, we're at the stage of trying to characterize what these patterns are. And so some of you may recognize this. This is the metabolic map of E. coli. And basically, it allows, basically talking about all the kinds of molecules that happen in metabolism in E. coli and how they react with one another, basically to keep us alive or to keep an E. coli alive. So I like doing a lot of analogies with physics, obviously, because I was trained in physics, and I think very much from the perspective of a theoretical physicist. So to talk about how I'm thinking about looking at those properties of that chemistry and what those emergent properties are, I just want to make an analogy for how we can think about characterizing physical systems. And statistical mechanics is a well-known theory in physics that basically describes emergent properties of many interacting particles. So if you think about temperature, temperature is an emergent property, right? It measures the average kinetic energy of particles in a room. So it's not a property of an individual molecule, it's a property of many molecules. And so in statistical mechanics, we don't really care about talking about the independent position and velocity of molecules in a box. We talk about their average properties, like their temperature. And then we can actually then talk about this box being hotter than this box, is comparing something about their statistical emergent properties. And the interesting thing about when we start doing that is we're no longer talking about a specific system, we're talking about what we call an ensemble of systems. Right? So we have this particular set of systems that all have the same temperature. They might have very different microstates, basically different positions and momenta for all the particles in the box, but on average they have the same temperature. And then you could say that this ensemble of boxes is basically hotter than this ensemble. The reason that this becomes important is that what we're interested in is basically maybe like the statistical mechanics equivalent of biochemistry. If you took all of biochemistry and you put it in a box, you wouldn't actually do the things that we do in physics where we just count the individual positions and momenta of the states. But the problem with doing this kind of thing is that molecules, as they react in biochemistry, actually have internal structure. And now it's not just the things that are present that matter, but the transformations between the things. And so chemistry is actually really interesting for doing statistics over because every molecule is really different. And so the way that people have started to talk about statistics or statistical mechanics in sets of things where you have very heterogeneous components and the transformations among the things or their interactions start to matter is with something called network theory. And so there's a whole literature on complex networks and statistical mechanics basically of complex networks. But the basic idea is now not just the individual elements matter, but how they interact with one another matters. And so you represent now the entities as nodes in these graphs, and then you connect them with edges. And so a really well-known example of how to treat things from a statistical standpoint with a network is to think about social systems. How many people here are on like Facebook or Twitter or something? Anyone? Some people, okay. And then you'll have friends in your social network. So you would be a node in a social network, every person in this room. And does anybody in here have like more than 500 friends? Just curious. On a social network? Okay, Shauna does. So she's a hub. How about other people? Do they have, yeah, how about like more than 100? Okay, how about less than 100? A few people, okay. So I just did some statistics over the network of social interactions in this room. Basically what people will do is they'll represent networks in that way. So Shauna would be like a node with lots and lots of connections because she has lots of friends. And other people might have fewer connections, and this is like me over here. I'm just having one friend. So what we can do is actually think about how we would do statistics over the interactions of molecules in kind of the same way. And you would connect them by an edge if they participated in a common reaction. And so this allows us to think statistically about biochemistry. And the analogy with thinking about statistical mechanics from the framework I gave before is that what we want in our box is living chemistry. And what we want in the other box is non-living chemistry. And these are actually representing ensembles. Right? But what I mean by ensembles is that if I want to look at the box, this boxes of living chemistry, I might take one organism as one box, another organism as another box, and another organism as another box, or an ecosystem as another box. I'm going to show you what our data set looks like in a minute. And I want to look at the statistics over all those boxes and say something statistically significant about why that set of boxes, the ensemble of biochemical networks, is different than non-biochemical networks. So how do you talk about the emergent properties of all of living chemistry as being distinct from non-living chemistry? And that's sort of the goal that we're going for. So that's the question. Is the ensemble of biochemical networks statistically distinguishable from non-living chemical networks? And you can see like where this would have implications for the origin of life transition, because if we could talk statistically in this sense, we might be able to figure out how to steer a non-living chemistry toward a living chemistry. And that's actually one of the goals of this work, is to really be able to address the origin of life question from a quantitative theoretical perspective. So what we do to actually do this kind of work quantitatively and think about these kind of mostly abstract philosophical concepts in real data is to take very large databases of biochemical networks to construct an ensemble. And so in our database currently that my group's working with, we have about 21,000 bacteria, genomes, 845 archaea, and 77 eukaryotes. We also have 5,500 metagenomes, so that's when you take an environmental sample and you just take the entire genomic content of the environmental sample, but you don't care about the individual species. So we call those ecosystem level networks. And then we take the biosphere as a whole as the 8,700 or so known enzymatically catalyzed reactions. So this is an ensemble of chemical reactions that are known to happen in biology. And then these are ensembles of ecosystem level networks and individual species level networks. And then what we do to actually construct those as networks is we take the genomic data and it's annotated by what enzymes are present or encoded in that genome, and we look up those enzymes in the KEGG database, the Kyoto Encyclopedia of Genes and Genomes, and we convert that into a reaction network that basically is a graphical representation of all the reactions that happen that organism can catalyze, or that ecosystem can catalyze. And so we end up with 21,000 plus networks for individuals in biochemistry, individual organisms, and what reactions they catalyze, and about 5,500 networks that represent the reactions that ecosystems can catalyze. And so the way I think about this is this, 21,000 examples of the set of chemical transformations an organism can do, and 5,500 examples of the set of chemical transformations an ecosystem can do. And then thinking about that biochemical, the set of all known biochemical reactions, obviously all the examples I just had on the previous slide are subsets of that, but you can also think about planetary scale organization of life as in what is the set of all known catalyzed reactions biochemistry can do, and create one network that's a planetary scale network for what biology controls chemically as far as transformations. So that's our data set. And now the question is there anything universal about the statistical properties of those systems that we could say is a regularity of biochemistry on Earth that might be a candidate for being something that could construct law-like principles for life on Earth and also life on other planets? So that's a question. So one way of asking that is to ask about scaling laws. Physicists are really infatuated with scaling laws, basically when one physical property scales with another. And so what we were interested in doing is actually looking at network size, so the number of compounds in the system, and whether any properties of that system scaled in a regular way with the size of that network. And so there were three different hypotheses that we actually wanted to test with that, looking for this idea of whether there's universal principles. One is that is the one that we're actually after, which is that there are scaling laws that describe individuals, ecosystems, and the biosphere that look the same across those three levels, and therefore are universal to biology, but they're different than random chemistry. And so this would be suggestive that those scaling laws are capturing regularities of living chemistries that aren't present in non-living chemistries, because there's a scaling behavior that we see in biology that we don't see in random chemistry. But in order to actually prove that this is the case, you'd have to have the two alternative hypotheses that you might also observe. One is that it's a universal property of chemistry. So maybe if we look across all those scales in biology, all those different ensembles of networks, we see the same scaling behavior. But if we did random chemical networks, some other kind of control, we'd see the same scaling behavior. So it's not something that's unique to transformations and the sets of transformations in biology. It's just a property of chemistry. And the other hypothesis is that it might just be that every set of systems we observe has different scaling behaviors. So individuals have one scaling behavior, ecosystems have another, and the biosphere as a whole is on neither of those scaling relations. And then random chemistry has something different. And this would suggest that there's really no universal properties. Right? So one of our arguments for looking at ecosystems and individuals is if we see the same regularities across scales, they're more likely to be universal than they would be if they were just universal in the sense astrobiology wants them to be universal than if they were just properties of individuals. And so these network properties, what are these kind of things? Well, the stuff that we were looking at is topology. So topology would be things like when we had the example of Shauna and I was talking about how many friends she has, that would be called her degree as a node. So you might look at the average number of friends people have. That's a topological property of the network because it relates the properties of nodes and edges. You might also look at clustering. So it would be like how many friends does Shauna have that I also have in common, and you can look at the connectivity of the patterns of the sets of interactions from that perspective. And we also looked to not just say that these were properties of networks, because network representations are really powerful mathematical tools, but they're highly dependent on the way you choose to represent your network. So we wanted some kind of control that wasn't dependent on our network representation. So we also looked at the diversity of reactions and enzymes and how they scaled. And so what we see is, in fact, that individuals and ecosystems have very regular behavior. So this is number of reactions, this is number of enzymes, this is number of compounds. The blue data is all 21,000 plus individual genomic level biochemical networks. So how many reactions they had versus compounds, how many enzymes they had versus compounds, and the red data is for ecosystems. And so basically we see very similar behavior for individuals and ecosystems as far as the structure of biochemical networks and the diversity of the reactions and the enzymes and how they scale as a function of system size. If we look at network properties, many of the network properties, but not all of them. So I'm showing some that did have regular scaling behavior, but some of them don't across levels. Average shortest path would be like if we had two nodes and we want to know the average path, or the shortest path between them, it would be 2 in this case because I can connect them by 2 units this way or 2 units this way. Whereas if I went a different route, it might be a longer path. So average shortest path basically counts the shortest path between nodes over the entire network. And clustering measures how many nodes in the graph have neighbors that are connected to each other. If we look at these topological properties, we see that they also scale in a really regular way across individuals and ecosystems. So not only does the diversity of reactions, but also the patterns of how the molecules are connected through those reactions have statistical regularities. One of the things that we were also interested in with this work is thinking about planetary scale networks, as I mentioned. So we actually looked at the set of compounds that are universal across life and how they contribute to network structure versus archaea, bacteria, and eukaryotes each have sets of compounds that are unique and specific to the different domains. And it turns out if you pull an individual archaea or bacteria or eukaryote and you measure just its topological properties and its size, you can predict with high statistical accuracy which domain it came from. Which is suggesting that the topological properties are actually playing into biological diversity, but they are also highly regular across all of these structures. So they're constrained by the scaling laws, but also statistically distinguishable, mostly because of the size of the networks. So if you recall, so basically what I've showed you so far suggests that all life on Earth, at least across scales for our data set, individuals, ecosystems have regularity in terms of reactions and the way those reactions are wired together statistically across all life, right? But we haven't proven that that's a unique feature of biochemistry yet. And so what we ended up trying to do was construct random networks that were of similar size to the biological networks and compare them to the biological networks by the same measures. And so this is, again, number of reactions versus number of enzymes. And this randomly sampled reactions is we took the global set, that 8,700 or so biochemical reactions that biology catalyzes, and we just randomly sampled them to make networks of a given size. When we do that, we see the orange scaling, which is close to biology, but not exactly the same. It's actually statistically distinguishable, so you could tell that this wasn't real biochemical networks. We then actually didn't sample those reactions randomly, but we sampled them by the frequency that they occur across living systems. So we know that all life on Earth shares a common set of reactions, and then the rest of the reactions are kind of more sparsely distributed. So we actually sampled based on that frequency distribution. So these networks that are constructed here in yellow are highly likely to have the same reactions as are common across all of life, but they're randomly selected from that frequency distribution. And that scaling behavior is much closer to the real scaling behavior that we see in life, which suggests that random chemical networks can be distinguished, even if they share the same global set of reactions. So you imagine if we moved outside that set, they'd be even more distinguishable. But ones that start to have the properties of having that common frequent set of reactions start to exhibit the same scaling behavior. So we do think that it's actually a product of the particular chemistry of life. We also randomly sampled networks at the level of genomes to make random ecosystem level networks composed only of archaea, bacteria, and eukaryotes to see how they contributed to the scaling. And those are mostly not distinguishable from the actual scaling behavior of real ecosystems, although you can, in some ways, tell the difference for some of the topological properties. And then the same thing with the topological measures. So in this case, for example, with clustering coefficient, you can see that the randomly sampled chemistries are very different, especially as you get to the biggest biological networks. But if you sample based on frequency, you start to see more convergence. And so random chemistry doesn't exhibit the same scaling behavior. You can statistically tell the difference between random chemistry and real biochemistry. But as you start to constrain the systems to look more biological, you start to lose that distinguishability. So basically explaining the scaling laws, which are regularities in the statistical properties across all of these biochemical systems, requires the individuals, the lowest level networks, to actually share the common set of reactions as the basic units. And so I'm going to start talking about some other work that our group has been doing. But one of the things that I thought was super interesting about this is that we have this kind of story of the last universal common ancestor and why we have a common set of reactions as an evolutionary story. But I think that this suggests something that might be really interesting and fundamentally different as a hypothesis, which has not been validated, but I'm really interested in testing, is whether the last universal common ancestor actually arose because of constraints on this global architecture of biochemistry. And that really the scale we should be thinking about is planetary scale biochemistry. And that individual organisms are subsets of that. They're sub-networks of that network. And that if we're thinking about the origin of life and the nucleation of life as a planetary process, which a lot of people in my field are starting to think about, that this planetary scale network is actually really relevant. And thinking about its global organization and its intersection with geochemistry, and that individuals might have then emerged out of that. And that's why we actually see the kind of universal properties of biochemistry that we see, which is very consistent with this idea of like the citric acid cycle being kind of primitive metabolism and those kind of thinking that people do. So the main conclusion from this work so far is the specific scaling relations observed across life emerge due to the destruction of interaction of compounds common across all life, and they're not a general characteristic of non-biological chemical networks. So you can tell the difference between core biochemistry and the biochemistry common across life statistically just by its structure, the interactions of the molecules as being distinct from non-living chemistry. So this leads into like the next set of work. So the rest of the stuff I'm going to present now is not published stuff, but stuff that we're working on writing papers on now, so it's a lot newer. And it's basically based on the whole framing of this thinking about planetary scale networks that we developed in the work that I just described. And so I want to go back to this idea about how we think about molecules and reactions leading to the emergent properties of living networks. And so one of the properties that I've been really interested in throughout my career, which I know people here have also worked a lot on, in particular Bob, is this idea of molecular chirality and thinking about the fact that life on Earth is homochiral. And what by that, it's meant that basically proteins are composed of left-handed amino acids only, and sugars in DNA and RNA are right-handed. Even though if you went like prebiotically and tried to actually look at the synthesis of them, you might get both kinds of stereoisomers because those molecules come with a mirror image form. So what we're interested in doing is kind of thinking about the problem of chirality in life slightly differently and not focusing on the homochirality of biopolymers, of these macromolecules, but what are the statistics of chirality in biochemical networks generally? And is that also a signature of life? So we took our same database that we have, where we have the 28,000 biochemical networks, and we labeled every compound that biochemistry has, whether it was chiral or not. So does it have a chiral center in the molecule or does it not have a chiral center? So this is something about the number of stereoisomers that the molecule has and something about the symmetry properties of the molecule. And if we actually look at the statistics of that, we can just count how many chiral compounds are there encoded in each genome or metagenome, and how does that scale with the total number of compounds. And it turns out if we look at the same data sets that we had, bacteria, archaea, eukarya ecosystems, they actually have very regular behavior in terms of the number of chiral compounds. And so it's almost a fixed ratio of around 65 to 70% chiral compounds. And if we look at the randomly sampled networks, they actually have a lower ratio. So this is not even looking obviously at random chemistry, it's just random biochemistry. But biology seems to maintain a sort of fixed percentage that's not shared by randomly sampled networks. So before I was looking at the statistical patterns of reactions, but now we're just looking at the statistical patterns of the properties of the molecules themselves. If we look at chirality across the reactions that are used by life, there's particular patterns in terms of the use of chiral compounds in reactions. So if we look at the KEGG database that we're using for cataloging biochemical reactions, reactions have certain labels on them, and then they have the compounds that are the reactants and the compounds that are the products. We can look at the chirality of the compounds that are used as reactants in the reaction versus the chirality compounds in the products. And we see certain patterns where biology favors certain conversion ratios of chiral to achiral or achiral to chiral. And so one of the things that we're really interested in is why biology is selecting out those particular transformations in molecular space. And we don't have an answer to that yet, but that's one of the kind of statistics that we're looking at. And we do think that this is actually necessary for biological function or has something to do with biological function. Because if you look at chirality across the evolutionary domains, we can actually see specific patterns that distinguish domain-specific reactions. So I showed that global network that had all the compounds universal to all life and then the compounds specific to each domain. We can also look at that in terms of reactions common to all life or reactions common to specific domains. And if we look at the reactions unique in each domain, we actually see that there are statistical differences in their use and conversion of compounds relative to their chirality, whether they're chiral or achiral, and how they convert between them. So if we universally look across all the compounds across all three domains, we see a particular pattern. And if we look at the fraction of compounds in each domain, there's clear differences in particular between archaea and bacteria, which suggests that there might be some really interesting early history with chirality with respect to how biochemical networks are structured. Another set of tools that we use is something called a network expansion algorithm. So if we think about this as like our biosphere level network, we can actually explore how biochemistry has navigated this chemical space doing something called network expansion, where we take that background network, we drop two compounds on it, we see how they react and what compounds they produce. And then through the next iteration of the algorithm, we react those compounds together and we get the next set of reactions. And then you can basically do a percolation over the chemical network to see how much of the network gets covered from any starting point. So this is a way of seeing how, if you start with a particular biochemical molecule, how you can move out into biochemical space. And so what we wanted to try to see is whether there was a difference between achiral and chiral compounds as far as their ability to lead to more expansive coverage of biochemical network space. The reason for that is if you think about achiral versus chiral molecules tend to be more complex. They have more kinds of transformations that can happen because they have different kinds of stereoisomers. So you might have the hypothesis that chirality is actually really important because of the diversity it brings to biochemistry in terms of the kind of transformations that can happen and their specificity. And so what we ended up seeing was that if we look at having different seed set size, so I just start with two random achiral molecules. If I look at the expanded network size, across biochemistry, most of the networks that you get are actually really small because most molecules are on the periphery of the network, but you might get a few that are kind of really critical to biochemistry and expand to large networks. And so this is just taking two achiral molecules and a random set of 10 achiral molecules, hundreds of sets of random samples of these molecules, and then looking at the distribution of network sizes. And what we see is if we randomly sample achiral molecules versus chiral molecules, we get really different distributions, similar kinds of sizes of networks selected, but if you look at the largest networks that end up being formed, you're much more likely to get to that domain of biochemistry if you start from a chiral precursor than if you start from an achiral one. So interestingly enough, we did this with like a prebiotic sample of compounds, which I'm not showing the data for, but if you start with prebiotic compounds, there's an interesting pattern about how chirality is added to the network as it expands as far as chiral compounds. And so one of our hypotheses is that early on, chiral compounds would have been really important to leading to massive expansion in biochemical diversity, and this is probably one of the reasons that chirality started to become important to life, even before we started to get like biopolymer chirality really mattering. And so one way to think about how we're thinking about the problem as far as origins of life is there's macromolecules and what's happening at the level of macromolecules, but there's all this small level systems messy chemistry that was happening underneath. And there had to be some symmetry breaking and some selectivity at both levels at the same time. And so we're studying the statistics of the lower level to understand how the higher level structures ended up emerging from that statistics. Okay, so this is gonna be the last part of my talk. How much time do I have? I know we started late. Okay, perfect. So I've been talking about understanding biochemistry on Earth and its regularities. And we've uncovered some clear statistical patterns both in the structure of reactions and also in the properties of molecules without any detailed knowledge of what those molecules are or what those reactions are. The ultimate goal of that is to be able to then take those statistics and be able to predict what biochemical networks on other worlds might look like, starting from the precursor chemistry. And so one of the things that we're doing is not predicting yet, because we don't know all the regularities and we haven't built the theoretical formalism, what biochemistry on other worlds could look like. But we can take biochemistry from our Earth and put it on other worlds and ask whether it would be even viable from our scenario, or what the space transformations from Earth's biochemistry might be possible on another planet. And so when we're thinking about this problem, we were actually really interested in doing that same network expansion algorithm I just talked about, but taking biochemistry from Earth and putting it on Enceladus. Because Enceladus, we have pretty well characterized what compounds are present on Enceladus, at least relative to other planets in our solar system, or moons in our solar system. And so one of the questions you could ask is how much of Earth's biochemistry could actually persist on Enceladus. Now, this is not including anything about thermodynamics or kinetics or anything like that, but just in the space of transformations, could you even get there from the starting condition of what molecules are available on Enceladus? And so what you can do is do that network expansion, whereas I started with achiral or chiral compounds before, now you can just start with the molecules you know are present on Enceladus, and then you can see how much of biochemistry actually could be produced in principle on Enceladus using the same kind of network expansion algorithms. And what we find is that if we look at that biosphere level network and we take Earth's biosphere as a set of chemical transformations and we just put that network on Enceladus, we can produce over 4,000 compounds. And so a lot of biochemistry on Earth would actually, in principle, be reachable in chemical space on Enceladus from those starting conditions based on known reactions. And so one question we were interested in is if we're not sure about what the molecules are that are available on Enceladus, or we want to understand how Enceladus conditions actually lead to viable biochemistry, we could actually sample smaller numbers of molecules from Enceladus and see how much of the network we actually end up getting. We sampled based on just not weighted for the concentration or abundance on Enceladus, so we just assume that everything is equally abundant on Enceladus, versus weighting by concentration. And part of the reason for doing this weighting is actually for probabilistic frameworks for biosignatures that I'm developing in a different context for exoplanets, which I'm happy to talk about later. But this is like assuming you didn't know the actual distribution on the planet, but you had some likelihood distribution. What would be the likelihood of products that you might get, assuming biochemistry like Earth? And it turns out that you can get most of the compounds with just 10 compounds from Enceladus. We also looked at the statistics over the different compounds that are available on Enceladus and how much of the biochemically expanded network you get. And if you always have water present in that seed set, you almost always get to the fully expanded network. But for other compounds, you're statistically less likely in combination with some of the other compounds. And so this is just sort of a model for thinking about how we can use this network theory to think about the viability of life on other planets, or what sets of chemical transformations could be present in other environments. Okay, so for the last part, I'm actually going to switch to not thinking about biochemistry as a complex system using this network representation, but planetary atmospheres. And the reason for that is that when we're thinking about detecting life on exoplanets, Our point of contact is going to be the atmosphere. And so just like you can think about patterns in chemical reactions in biochemistry as a statistical regularity that you might say is the signature of life, if you think life is a planetary scale process and it's driving atmospheric processes, there should also be signatures of life in the statistics and the patterns of interactions of molecules in atmospheres. And so this is sort of the idea that we have in mind. And part of this is that we can think about the biospheres as complex planetary scale network, but we could also think about other planetary scale systems as networks, which people here obviously do, thinking about minerals as networks. But if you can imagine thinking about the atmosphere as a network, the biosphere as a network, geochemistry as a network, minerals as a network, then you can talk about this multi-layer, huge data structure that allows you to understand the statistical regularities in the interaction of those subsystems. So this becomes an incredibly powerful set of mathematical tools for understanding these interacting systems in ways that we haven't understood in the past, that allows us to naturally think about their complexity and also think about the biosphere's role in driving these other planetary systems. And so we've been particularly interested in what is the network structure of atmospheres. And the next step for us is going to be to couple atmosphere networks to biochemical networks to understand how those two systems are interacting. And so just to go back to some of the stuff I was talking about earlier before, the basic idea is how do we distinguish a non-living and a living world? And for a lot of astrobiology, this is kind of a stab in the dark in some sense, because so far we've only been able to look for Earth-like life on Earth-like world, but there's a whole diversity of exoplanets we're finding, and potentially an entire diversity of life. And so we don't want to just be anthropocentric in the way that we're approaching the problem. And so the network perspective is allowing us to think more about patterns and statistical regularities rather than specific molecules that might be indicative of life on Earth. And once we can actually couple those systems and understand the coupling of, say, the atmosphere to the biospheres and the regularities in those systems, you can presumably transport that to other chemistries and other planetary compositions. And so the idea is if we take the atmosphere of a few different planets and we look at that from a network representation, and here's just shown Mars, Earth, and Venus, Earth's network presumably should look different because of the presence of biology. And in fact, complex system scientists, because they like to make graphical representations of lots of different systems, actually did some analysis in the early 2000s comparing the atmospheres of solar systems in our planet, in our solar system, from this network perspective, and show that Earth's network is much more heterogeneous and has a structure much more like what we see in biochemical networks than the other atmospheres in our solar system. And so there is some preliminary evidence that this kind of approach might actually pick up the fact that Earth's atmosphere is complex in a way that's driven by life than other atmospheres. But this requires a lot more validation, because anybody in here that knows anything about atmospheric science will know that these reaction lists are not very tightly constrained, and so a lot of people will add reactions to certain models or not other models. And so the reaction list that might have specifically been used for this comparison might not be truly representative of the reactions happening in that atmosphere, because we don't know the exact reactions. And so this becomes one of the biggest problems, not just for this kind of network representation, but exoplanet science in general, or any kind of planetary atmospheric science. So we don't know the exact models. We don't know the exact set of reactions. One of the reasons I like the network approach is that when you look at the aggregate statistics over these network representations, they're very insensitive to whether certain reactions are present or not. They tend to be very robust. Particularly certain kinds of models can be more robust than others, so you're not going to change the overall structure of this network necessarily by adding one reaction or moving one reaction. So they're a really robust statistical representation of atmospheres, but more importantly, to the point that we don't know the exact models for exoplanets, one of the things that we really need to start doing in exoplanet science, I think, is moving to more statistical frameworks in general. And so there was actually a series of white papers that came out in astrobiology last June, I think it was, on future directions in exoplanet biosignatures, reviewing the field and also setting, what's the next phase of research for exoplanets. And this part of the figure actually comes, without the networks, from the intro to that set of papers. I led the paper on future directions in exoplanet biosignatures, and the main premise of that paper was basically the fact that when we look at distant exoplanets, we are going to have extremely limited data, like a pixel of data, a little bit of a characterization of the atmosphere, and then how do we infer the presence of life from that? And so the idea of that set of papers was that we need some kind of statistical framework or Bayesian framework to actually infer what the properties of that planet are based on that limited data. And so right now, where people are starting to really do that is with the class of models called hot Jupiters. And so what we're doing is actually working with a collaborator that does modeling of hot Jupiters based on observational constraints. And so the methodology there is you get some observation of a planet, you do some Bayesian retrieval methods, you build a class of models that's the most likely set of models for that atmosphere. And so that would be like a class of kinetic models. And then what we're doing is actually doing network representations of those models and trying to understand whether the network theory actually can track any of the properties of the atmosphere that we think might be relevant to eventual life detection. And the thing that we're focusing on now is disequilibria in the atmosphere. With the eventual goal of going to limited data sets for terrestrial worlds and trying to infer likelihood distributions of models for those atmospheres and determine whether that distribution of models for a living planet is distinguishable from the distribution of models for a non-living planet. Right? So it's a statistical distinguishability. So how do we go from observations to atmospheric networks? Well, the idea is, and this is with Mike Line at ASU, he gets some atmospheric spectra, he does this retrieval model, he gets molecular abundances, and then kinetic model constraints, and he basically can build a set of the most likely models for that atmospheric spectra. And then what we do is we basically take that set of most likely models, say if they're fixed with some particular temperature, and then we do network topology and statistics on those models to build distributions of the network properties that are consistent with that atmospheric spectra. And what we're trying to do right now is actually look at disequilibria in the atmosphere. So this KZZ is a measure of vertical mixing. And what we're finding is that basically if we look at vertical mixing strength, so if it's 0, it means that the atmosphere is at equilibrium with respect to mixing. And then if it has larger values, it means that there's more vertical mixing happening. And we're looking at network properties like mean degree that we saw before, average, shortest path length, or clustering coefficient, and just seeing if they actually track the disequilibria in the atmosphere and how they track it. And so this is a way of saying, so for the equilibrium distribution centered about a temperature of 1200 Kelvin for this particular exoplanet, this is the distribution of values we would see for the mean degree of the reactions in that atmosphere. And then these are the non-equilibrium distributions that we would see. And so you can see that the network theory actually tracks the disequilibria, but in different ways for different measures. So this provides like a statistical database, basically of characterizing the properties of the atmospheric chemistry with different disequilibria drivers. And so what we're hoping to do with this eventually is use this as a way of understanding what the signatures of living disequilibria would be in planetary atmospheres from this network perspective. So that's really new work. And just to kind of like bring my talk back around, basically the whole idea of like the entire talk was that life is an emergent property and we should understand its statistical regularities in order to be able to infer what kind of law-like properties might govern living systems. And so if we're thinking about life as emergent property, it has to arise from the interactions of many molecules, and therefore its properties should be embedded in the statistics of those interactions, which is why we're choosing to use network theory to understand it. And so this is kind of a new mathematical framework for astrobiology that's based on big data and looking at statistical regularities that we can apply across all chemical systems and try to start coupling those systems and also couple to other kind of network representations like the mineral networks that you guys are studying. But I always go back to this, especially with the beginning of my talk, that anytime I think about life, or life as a planetary scale process, or life as any process, that I still think that there is a lot missing in our descriptions of the natural world. And I just want to thank my sponsors and my amazing lab. Hanju Kim and Harrison Smith did a lot of the scaling work along with Cole. And the chirality stuff, Hanju's been leading with John, who's not pictured, who's one of my newer grad students. And Tessa here has been doing the atmospheric network stuff. And with that, I'll thank you all.
Sarah, that was really inspiring. Okay, so who has the first question? All right. And we'll use the mic because we're recording.
So while you were talking, I was thinking really hard about ribosomes and trying to figure out how one can statistically distinguish between coding and non-coding regions of DNA, and how you do that in terms of reaction systems rather than the actual pattern of DNA itself. So have you taken a look at that, or is that?
No, but that's a really neat idea. Yeah. I guess I wouldn't. I think that you could look at it in terms of the pattern. You mean in terms of the patterns of the reactions to construct the element of like non-coding or coding DNA? Oh, I see. Okay. Yeah. Yeah. Right. Yeah. Yeah. You should in principle see clear differences in that, but I haven't thought about looking at that. But that would be an interesting case study. Yeah, that's cool. Thank you.
Hey, Sarah, I'm Fabo. Hopefully you'll remember me.
Yes, I do.
Yeah.
Nice to see you.
Okay, nice to see you. So, yeah, you mentioned information, and I think life is related to information, and probably two different networks, the biotic and abiotic networks. So you didn't mention why, how the biotic network emerged from a biotic network and how the information emerged from some kind of non-informative.
Yeah, I mean, I think that's the ultimate question. So I'm obviously most interested in that question, but part of the thing that I've had to do is kind of take a step back from that because we don't even know what it means. So this actually, like the trajectory of my career was like I started my postdoc doing modeling of like origin of life, like prebiotic chemistry. And it became like very obvious very early on that I had no clue what I was doing because there were no metrics for what a living network was. Right, so it's almost impossible to ask the question of how do you like talk about the transition from non-life to life if you don't know what life is. And for me, being a quantitative scientist, I want quantifiable metrics, not just descriptors that like we have now. So I think that there are ways of starting from like the approach we're doing to apply that to like prebiotic chemistries and start to think about when do they become more lifelike and how could you steer them to be more lifelike. And one of the projects I am working on is with a collaborator, Lee Crone at University of Glasgow, and he has these like mineral steering reactions. So he takes these complex, messy chemistries and introduces minerals in particular orders and sees kind of that you can get statistically distinguishable product ensembles. And so one of the things that we're trying to do eventually is use the kind of statistical regularities that we're looking at in those kinds of systems to talk about when they start to have more lifelike attributes from just, like what's the distribution of molecules, what's the distribution of reactions. And then you could imagine trying to figure out what sequences of environmental factors might lead to more lifelike chemistries. So that's one way of approaching it. But I think in general there's, this is sort of my long answer. So that's my sort of like practical, what could you do in the lab kind of thing. And then there's my more theoretical abstract perspective on it, which is that I don't think that we model chemistry the right way to capture the physics. And part of the reason is that like if you look at origin of life models, people always treat individual molecules as like a node. So if you look at like the way people talk about autocatalytic networks, for example, a molecule is a node, it has no internal structure. It doesn't matter what atoms it's composed of, and the transformations on those atoms then therefore don't matter. There's some things like graph grammars and stuff that start to get at that, but I think we have to start thinking about molecules as objects and looking at how they transform into each other and then understanding the physics of how matter flows through that space of objects to understand how life emerges. And that's actually where information comes in, is when you think about molecular diversity, there probably is a certain scale in chemistry where all the things that are possible can no longer exist physically in the universe because there's not enough stuff to make them. And Stu Kaufman talks about that quite a bit as far as like the adjacent possible, and that you just get into the space of things that's too large to actually exist. And that's where information actually starts to matter because you actually have the information to specify the objects that do exist. And so there is something interesting about thinking about what molecular space is and how information is necessary to specify objects in that space that would probably form the relevant physics. That's my long answer.
Thank you. Another question is about, so you mean networks, but you didn't mean, whether the environment can perturb the networks, whether it is robust to the perturbation or not. I'm not sure you consider this kind of factors.
Yeah, so we had to do some robustness studies just to prove that the scaling laws weren't sensitive to the fact that we were missing data, because genomic data is never really complete. And so they are pretty robust. You can knock out like 10 to 15% of nodes and still see the same scaling behavior. So this is another thing about the network representations being good ones that are sort of nice when you have limited or incomplete data, because they only care about the general statistical properties, and lots of the details then don't matter as much, which is one of the reasons I think it's a really useful tool for astrobiology. So from that perspective, we looked at robustness, but not necessarily to like big environmental perturbations. And one of the things I really want to do is not just do this with biochemical space, but chemical space more generally, and understand how biochemistry is embedded in chemical space. But that requires some of the theoretical tools that I was just mentioning that we're trying to develop now, but it's going to be a few years.
Thank you.
Sara, so when you look at the metabolic map of E. coli, there's the statistics that you can pull out of that.
Yeah.
But then I think the other way to look at it is to think about the statistics of the frequency of any given reaction.
Yes, yeah.
So
we're
I
gonna
have a
do that,
figure
yeah.
that I'll show you later today, of what I call an NMR's spectroscopist's view of E. coli. And it's remarkably simple system. And so it shows you that there's a very non-random frequency distribution. And I think that it would be fascinating. You know, certainly if you just allowed everything to be equally probable, the molecular distribution would be so far from what E. coli is. It's an interesting thing to look at.
Yeah,
That's just a comment, I guess.
Yeah, no, that's a great comment. Yeah, so this has nothing to say about abundances or what molecules exist in what environments and any of these kind of things. And I go back and forth with my group about that all the time, but I'm super interested in sort of the abundance distributions also. But the argument I make for this is that as I said through the whole talk, one of the things biology does is control the transformations that happen. And so I'm interested in those statistics, but that doesn't mean that all those things are actually present at a given time or matter as much to the real system at a given time. And that's sort of like a different dimensionality of the problem, which eventually those two probably, yeah, exactly, yeah, yeah. So that's cool. I can't wait to see that. I'm so excited.
Yeah, I'm a couple of things. I'm struck by you say you use a random network as a control, basically, against the biology. But at some point in time, it went abiotic, prebiotic, biotic transition, right?
Right.
So it implies within that transition that is non-random before life started.
Right.
So I was interested to see how your network theory could enable our understanding of that transition.
Yeah. Yeah, so I think that's a great way of asking it. So one of the reasons we did the random networks we did is just because we didn't have access to databases that had larger sets of chemical reactions outside of biochemical space. So we just randomly sampled within that space. Just as a proof of concept, biology was different than random in terms of its organizational patterns. But ideally what you would do is start from like, known prebiotic networks and all chemical available data, sample networks, look at their statistical properties and study the transition from things that we know are really not alive to things that are alive. And maybe over evolutionary history also.
I agree. And what you have to also think about is there is a feedback network. There must be.
Yes.
As one thing happens,
right.
it causes a feedback that
right.
produces. And that feedback loop is potentially one of the most important things.
And that feedback loop presumably leads to the structure we see, because we're not talking, I don't have anything about the enzymes in our network. We just assume the enzymes are there and they're catalyzing the reactions. But you could do a multi-layer network with the enzymes also present and then actually look at the interaction structure.
Even at
and
the
so
catalytic,
that's also a direction.
Yeah, even at the catalytic level, there must be a feedback loop. Also, in your description of physics, it's classical physics you're talking about and not quantum physics. I was wondering, do you have any thoughts on the latest results showing that a lot of biophysics could be quantum effects, like photosynthesis?
Yeah, so we just had a workshop at the Beyond Center on quantum biology. We've had a couple of them over the years, but our most recent one was in November. So we had a lot of the people that work on non-trivial quantum effects in biology at that workshop. My perspective on it is that life is not foundationally quantum. Life is weird, but it's not quantum weird. It's just probabilistically weird. And quantum mechanics is also probabilistically weird. So it looks like their physics is similar, but they're different. And the thing that I like to point out about quantum mechanics is we think that's weird that biology uses that, because we think quantum mechanics is so weird and it has a special ontological status in our theories of the world. But biology uses chemotaxis. It has knowledge of gravitation. It uses a lot of different physical effects. And what it does is it harnesses them and controls them. I think the fact that biology uses quantum mechanics isn't any weirder than the fact that biology responds to gravitational fields or chemotaxis. I just think that biology has information about how physics works and it controls it, and it could be quantum effects or it could be any other physical effect. Yeah, exactly. Yeah, it takes advantage of it. Yeah, so that's my personal perspective on it, is that the interesting thing about biology is that it controls physical processes, and it doesn't, yeah, I'll just stop there.
So
Yeah.
thanks very much. That was really inspiring. So all of these organic molecular systems are very information rich in a Shannon sense. What seems to me in biology is really important is that you're selecting for function.
Yes.
So I think the function is important. And what really struck me also in terms of the chirality distribution, the fact is you see a higher percentage of chiral molecules in the biological system, which suggests that, again,
Related to
there is
function,
some
yeah.
very critical set of functions that's associated with those molecules. So,
Yeah.
so have you thought about taking the networks a little farther and thinking about this idea of the functional information that's present and how that may be one of the keys?
Yeah, yeah. So 100% yes. And I think one of the things is eventually, I think there will be some information theoretic representation of our networks. I think the way that we're representing them now is a good mathematical approximation, but it's not the right way of representing them to do proper physics, as I would say, and that you would need some kind of way about thinking about molecules as informational objects, which could be a kind of functional information, and that would be the proper representation. So we are thinking about ways about doing that. And one of the ways that I really like to think about is using this pathway assembly measure that Lee Cronin came up with, which we're trying to help mathematically formalize. But that's basically about like the number of ways that you could make a molecule. And like what's the shortest sequence of steps to produce that molecule, which has something to do with about the pathways or the kinds of things that would be necessarily construct that molecule. So it captures something about a functional pathway. But I know there's other measures. I remember reading in great depth your paper, which actually was second functional information when I was a postdoc. I thought that was really great. And the other thing that we're doing is labeling our networks also by their function and pathways. And that was something that I think my group was talking last week with you guys out in the records group about doing better, because we've just been using the KEGG data, but they seem to have a better way of labeling that. So yeah, so definitely, yeah. So lots of things, yeah. So that one will be really exciting, yeah.
So have you thought about how you use this sort of the network theory to think a little bit more about evolutionary components here? Because I mean, you know, there's, you know, you're sampling obviously the Earth at a particular time in history and using that data for all this, but presumably there could be interesting parts in the evolutionary train where there's disruptive changes. And especially if you're looking at an exoplanet, is the temporal component. So have you thought about how you use that aspect to this?
Yeah,
this way of
100% yes. We haven't done it yet, but we have thought a lot about. So like I'm really interested in this biosphere level network, but I'm also interested in how that changed through time and thinking about that coupled to other networks. So ultimately, what I really like to do is about thinking about these planetary scale networks as a multi-level process and looking at some evolutionary model for how they co-evolved in time. And one of the things I think is really useful about the network theory stuff is if you talk about that as a set of constraints on the structure of those systems, you don't have to consider that to be a unique history, but you could talk about all the histories that are statistically consistent with the kind of observational data you have, which would allow you to produce not only like different trajectories for exoplanets, but also alternative trajectories for the history of Earth.
Yeah, but
so
even on Earth, it would be interesting to, if you go back in history and see.
I really want to do that.
Biology made some choices. It may not have always been the best choice, right? But it'd be interesting to see how biology makes those choices.
Yeah. Yeah, so yes, I'm very interested in that.
Okay, do we have another question? Okay, if not, let's thank our speaker. Thank you so much, Sarah. And we're having lunch after, so please join us.